Hey everybody, I just wanted to got a little bit of a video about the photosynthesis content that we were talking about yesterday. If your class was on Monday and Wednesday, you already got all of this and we made it all the way through the presentation. If you were in my Tuesday, Thursday group though, we did not because we had an earthquake drill. And so I wanted to kind of go through a little bit of this to make sure that you don't feel time crunched. We will talk about this in class on Tuesday to finish up if you are in my Tuesday, Thursday section. But since the exam is on Thursday, I thought I would kind of pick this out with the weekend to spare in case you wanted to catch up. This is our photosynthetic equation. So when plants are doing photosynthesis, this is what we get. Photosynthesis is a reaction. We can think of it as a biological process, but at the end of the day, it's a chemical reaction just like everything else is. So we have reactants and we have products. The reactants are carbon dioxide, 6 of them, and 12 molecules of water. This is all we're going to need chemistry-wise in order to actually get the products that we need. The product that we end up with is a glucose molecule. You can see it here, C6H12O6. So here's our sugar that we're going to make, one of them. 6 molecules of water, sorry, 6 molecules of oxygen. So as you know, oxygen is a byproduct of photosynthesis. We think about this colloquially when we think about trees and what they do. Oxygen comes out and 6 molecules of water. Now, I wanted to mention this because here we have 12 molecules of water going in and 6 molecules of water coming out. These 6 waters coming out are not 6 of these 12s that go in. So these 12 waters that go in get completely deconstructed, completely taken apart. And in the reaction process as it progresses, these 6 molecules of water get assembled in. So these 6 are not just 6 of these 12, right? These 12 completely taken apart, these 6 completely constructed, right, during the product. So what we want to do briefly here, we want to talk about how this happens. What is the biology behind this reaction and where does it happen? And what does this have to do with the green color and light and all that kind of stuff that we sort of associate with plants and with photosynthesis? So banging through here. Let me go on. Cool. So this happens inside of the plants, obviously, as we sort of already know, but more specifically, it happens inside of the chloroplast. So when we were looking at our lodia leaves, the chloroplasts with little green things that we saw trucking around inside of those cells, they're easy to see. Here is an Alodia leaf under a microscope that I took this picture in lab, right? So this is the same kind of stuff that we've been looking at all along. But when you think about the structure of a plant leaf and what it is doing, the chloroplast obviously is part of a much larger whole, obviously. So you can see the cells. Within the cells, we have the entire leaf. Within the leaf, we have the whole plant. So there's a lot of different levels of structure here that are important to us as we sort of go through. But even within the chloroplast itself, there are some structures that we need to pay attention to as we really zoom in. Looking inside of the individual chloroplast, we have these little structures that look like little frisbees, kind of stacked up, or like a stack of plates kind of piled up. Each one of these little components of this stack is called a thylakoid. A thylakoid. And it is on the surface of the thylakoid. That's where the chlorophyll is. That's where these light reactions of photosynthesis happen, right? So that's where a lot of this reaction actually takes place. So we're in the leaf, in a cell, in a chloroplast, on the surface of a thylakoid. That's where we're actually doing this kind of stuff. You can't see the thylakoids at this level. When we look at this up close, you can see the chloroplast, but kind of. You can see the chloroplast, but thylakoid is even smaller than that. So you got to really zoom in good to see those. As we go forward, all right, here's our chlorophyll molecule. It's really big. It's got a magnesium ion kind of tucked into the middle of it. This large structure on the top is referred to as a porphyrin ring. A porphyrin ring. And this is actually the same ring-like structure that we make heme groups out of that carries oxygen in your blood. So if you swap out this magnesium and put an ion of iron in it, then it will have a reddish appearance. But the magnesium with this chemical structure gives it the green. That's how we capture the electrons that we're going to use to make photosynthesis happen. So this is the stuff that's kind of embedded within the surface of the thylakoid. When we think about photosynthesis, we think about it in terms of two different kinds of reactions. So we have the light reactions or the light-dependent reactions. This is the part that happens on the surface of those thylakoids that you can kind of see here. And that is the part where we need the sunlight. It is the first thing that we do when we do photosynthesis. Sunlight is absorbed by the chlorophyll molecules and the energy is pulled out of it to start doing its thing. Here you can see the light kind of coming in on these light reactions. That's also where we use the water. So the water as a reactant is one of the very first things that we sort of use as we get kind of going. This is where we harvest energy. It's also kind of referred to as the energy harvesting steps. This is where all the energy that we're going to need to make this happen goes into the system, right? In these light reactions. Then we have this other set of reactions called the dark reactions, or sometimes called the light independent reactions. It's in these light independent reactions here. That is where we actually assemble the sugar molecules. So we take the energy that we get in the light reactions and we put them together into these sugar molecules. So this is where we do the construction of the sugar. And because it's called dark reactions, light independent, obviously we don't need light to do that. We're done with light after the light reactions happen. This is solely a construction process. This does not happen in the thylakoid, right? This happens just in what's called the stroma. It is the gooey, liquidy stuff, right, that is in the chloroplast that is not part of the thylakoid. That can happen 24 hours a day, not just when the sun is out. So that's cool. All right, so moving on, I wanted to show this slide here because this actually is the nitty gritty of the light reactions. So there's a lot of moving parts here on this slide, and this is really kind of the one that I wanted to show to really sort of bring it home as to what we're talking about here. So let me blow this up. All right, so the first step in our photosynthetic process is actually called Photosystem II. This might be a little bit counterintuitive. You would think it would be Photosystem I. It is not, right? The photosystems, there's two of them, Photosystem II and Photosystem I. They are named after their order of discovery, not their order of events. So even though Photosystem II happens first in the photosynthetic process, it was the second process actually identified, discovered, and described. So it was named Photosystem II. Goodness me. Anyway, so a lot happens in Photosystem II, as you can see from all the arrows and all the parts kind of plugging through here. The very first thing that happens is that a water molecule is split into two. So now you take a water molecule, you split it apart, you take the oxygen, and it goes off into the environment. That's where the oxygen comes from that we associate with the byproduct of photosynthesis. And you get two hydrogen ions. And those hydrogen ions that come off then just kind of build up here inside the thylakoid. If you remember, we are inside the thylakoid at this point. We are on the surface of it. That is where photosynthesis happens, and this is the process. So this right here is the wall of the thylakoid. So this is one of our plate-like frisbees that we have in our stack. So water is split, oxygen and hydrogen is released to the environment. The oxygen, since it's small and nonpolar, can just go ahead and diffuse out. The hydrogen ions, however, being charged, are stuck inside the thylakoid, and we're going to rely on that later. What we're then going to do, we're going to take the electrons out of it. So as we broke those covalent bonds, we're able to gather up some electrons. And we are then going to take these electrons, you see them right here, we are going to shine some light energy on them. All right, so the photon, the unit of light energy that we're going to be working with here in photosynthesis, is sort of the currency of the electron. So electrons can gain energy by absorbing photons. They release energy by releasing, or they lower energy by releasing photons out of them. So this light energy is going to come in, it's going to hit this photon, it's going to go ahead and absorb that. So now we have two high energy electrons. So broke water apart, put the contents out into the environment, the oxygen and the hydrogen, grab the electrons absorb the photon. Now we have high energy electrons and we're off to the next step. Moving over, we pass those electrons down to what's called an electron transfer chain or electron transport chain. What this is, it's a series of enzymes, and we are going to pass that electron from one down to the next enzyme down the line. As we do so, one of those enzymes is what we call a uniporter. In this case, specifically, it is a proton transporter. So what it's going to do, it's going to take some of the energy out of those electrons that we put in the previous step, and it's going to use that energy to actively, against concentration gradient, pump protons or hydrogen ions, they're interchangeable, into the inside of the thylakoid. So the more energized electrons we send down this uniporter, the higher our hydrogen ion concentration is going to get. So now what we have left is some hydrogen ions that are now inside of the thylakoid, and we're back to having some low energy electrons because we just took the energy back out of them to make that hydrogen ion gradient. The next step down the line is photosystem 1. Just like in photosystem 2, we're going to shine some light on this thing. Some photons of light are going to be absorbed by the electrons. That means we have these low energy electrons, light comes in, we're going to push these electrons back up to a high energy level, re-energize these electrons, as you will, here in photosystem 1. We are then going to put these electrons, and we're going to send them over to the next part where we are going to use them as the basis of a covalent bond between a carrier molecule. It's called NADP, nicotinamide adenine dinucleotide phosphate. And what we are then going to do is use that pair of electrons to form that covalent bond between the NADP, nicotinamide adenine dinucleotide phosphate, and a hydrogen ion. So we're using the hydrogen ion to essentially be the carrier of those high energy electrons. So what we now have, the electrons that we originally got out of water, here we've energized them, released the energy out to make a hydrogen ion gradient, re-energized, and now we're dishing them off and putting them into a bucket, essentially called NADPH. We are then going to take the NADPH and send it over to the dark reactions, send it over to what we call the Calvin-Benson cycle. We're not quite done. We got one more step. The next thing we're going to do, we are going to take that hydrogen ion gradient that we built here right after photosystem 2, because you remember now we have a lot of hydrogen ions inside this thylakoid. We're going to take these hydrogen ions and we are going to let them pass through this channel protein called ATP synthase. When these hydrogen ions pass through the channel, that is going to cause the ATP synthase to spin around. And as it does so, it takes a molecule of ADP and an organic phosphate functional group, and it binds them together to make a molecule of ATP. So that is also where some of our sunlight energy is. If you remember, the sunlight energy of photosystem 2 was put into making that gradient. We then release that gradient to make ATP out of it. We re-energize in photosystem 1 with more sunlight energy, and that sunlight energy goes into the molecule of NADPH. What we are then left with, that's where our energy is. It's in this ATP, it's in this NADPH. We send that energy then in the form of those molecules over to the dark reactions. So this is where we do assembly. This is where we assemble our sugar molecule. You can see kind of right down here in the bottom, this is where our sugar molecule is, right by my face. Here's our CO2, finally using that. This is the first time we've actually used the CO2. CO2 is coming in to the reaction right here, and it is being picked up by an enzyme called rubisco. And that rubisco enzyme is going to take that CO2 and it's going to take some of this 5-carbon chain molecule called RUBP, and it's going to combine them together to make the 6-carbon intermediate molecule. That 6-carbon intermediate is then going to immediately break apart into 2 3-carbon molecules. That's when we're going to start plugging in a bunch of ATP with it, and we're going to start plugging in and dishing off those high energy electrons from NADPH with it. So we use a lot of ATP and NADPH here on these 3-carbon intermediates to make this molecule called PGAL. And then we split, right? We take 2 of those PGALs down here, we combine them together to make a molecule of glucose. That's our sugar ultimate product that we get, and we've done what our photosynthetic thing is, almost. We take the rest of the PGAL that we've made, and we need to use it to keep the cycle going. So we have to take that PGAL, react it with some more ATP to replenish our RUBP. It's called ribulose biphosphate. We have to replenish that in order to keep the cycle going because eventually we're going to react our RUBP with our CO2. So the cycle is not a cycle. If it stops halfway through, we need to spend even more carbon and spend even more energy from ATP to make our RUBP to keep it going again so our Ribisco can react more CO2 with it to make more sugars. And those are our dark reactions. So altogether, we have done photosynthesis. We've done the thing. So we have harvested light energy by splitting water, taking the electrons, reacting it with the sunlight photons to make high energy molecules here, send those high energy molecules over to the dark reactions where we use that energy to assemble glucose and to keep our cycle going. And that is photosynthesis in a nutshell. So it's a two-stage process, light and dark reactions. The light reactions require the photons, the dark require the CO2. Together as a two-stage process, we get a molecule of glucose and we keep our cycle going so we can keep doing it again. So that's photosynthesis in a nutshell. I hope you enjoyed this video. I hope it was helpful. And take care. I'll see you guys soon. Let me know if you have any questions.